Hi, this is Dr. Jabali, and in this video, we're going to tackle some of the multiple choice for topic 2.8, which is cell respiration. So let's go ahead. Here it says which gas produces most of the bubbles in bread dough. So this is knowledge based. You have to be able to go and through cell respiration, specifically in fermentation, in order to be able to answer this question. So if you remember from pyruvate, right, after glycolysis, pyruvate, if it doesn't have what it wants, for instance, it doesn't have oxygen in this case, then pyruvate will go towards fermentation. And depending on which organism is it going, is it a human, is it a bacteria, is it a yeast, whatever it is going to produce a certain type of material from pyruvate. Now, in that fermentation, when it comes to bread, for instance, what is actually going to produce is CO2 because the bread, for instance, usually is flat like this, but then as the yeast is breaking down the bread, it's causing it to basically become to rise. So then the bread looks this thick instead of just being a flat thing. So the right answer here, it is C, it's carbon dioxide. Now, you need to know this more likely because, again, when we're talking about bread dough, we're talking about fermentation, and we know the fermentation has no oxygen, so this is already canceled. And again, methane, it's not something that we use in our body. For instance, we don't eat it on it. It's basically in the air. And water vapor, in the case, will not be the only reason to make a bread go up. So obviously, CO2 is the right answer. But again, this question was more of you having knowledge to study the information. Now, the next one. Which pair of molecules are products of the aerobic and anaerobic cell respiration in some organism? So we are talking about the products. Okay, this is asking for the products. And of who? There's two. There is aerobic here, right? And there's the anaerobic. Now, what exactly is each one of these? Aerobic is the one that has the oxygen. Anaerobic is the one that doesn't have the oxygen. So what are they asking for is the pair of molecules. And we're talking about their products. That means the end of it. Now, in the end of it, we always have to think, what exactly is aerobic for? Aerobic is in the end is about producing ATP, right? Anaerobic in the end, it can be about CO2 as well as NADH, and sometimes it can be ethanol, because these guys is all about fermentation. So, looking at this, aerobic oxygen, that's not a product. Oxygen here is actually a reactant, you're using it. And anaerobic cell pyruvate is also basically a reactant. These guys are not it. These are not products, so it's wrong. Second, aerobic cell respiration. Lactate. Lactic acid is something that is a fermentation that happens in humans where they don't have enough oxygen, so it's already wrong. And ATP, there's not much production, but it's still fine. But just because the B is wrong, this section is wrong, B is wrong. C, anaerobic cell respiration, CO2, could it be a product? Yes, it could, because when we do cell respiration, we're basically breathing out CO2 as we're breathing oxygen. However, glucose is not a product here because in any anaerobic, both, you're breaking down glucose. So glucose is a reactant, so it's definitely not it. Now, adenosine triphosphate is the same thing as ATP. Now, that is a product of a cell respiration because cell respiration is all about breaking down glucose to get the energy. And if you don't have oxygen, then you're going to release CO2, of course, by breaking down through fermentation. So the right answer here, it is D. Now, how do I know this information? Of course, you have to remember and study all about aerobic and anaerobic, knowing that one is with oxygen. If you have oxygen, you're definitely going to go through the Krebs cycle and you're going to go through the ETC in order to produce a lot of ATPs. If you don't have oxygen, then you don't do any Krebs or ATC. You go straight from glycolysis to fermentation. Okay, so this is basic knowledge-based information. Next, the graph shows the change in lactate. So there's lactate and pyruvate. Now, if you remember here, is that after glycolysis, right, which glycolysis, it basically means the breakdown of Of glucose. And what's end up breaking down glucose into pyruvate. So this is what it is. Now, to get pyruvate to become a lactate, that means there shouldn't be no oxygen at all. Oxygen should not be there. So let's see here. This was just a little scoop. Like when you read this, you start to remember, oh yeah, it's true, pyruvate is turned into lactate because there's no oxygen, so it's fermentation. So now you read the question, you already know what's the concept about. So let's read the rest. So it's measured in an athlete blood. So it's an athlete blood during and following a mild exercise period compared to before the exercise. So basically we're talking about after and before the exercise. So if you see the highlight here yellow, this area here is where it's actually the exercise is happening. So you can see from the graph, right? Now look at the graph. We have here the time in which the exercise is happening, and here is the relative concentration. Now concentration of who? We look at the key here. We have lactate and pyruvate, okay? So just highlight, I'm going to take the highlights just for you to continue seeing these numbers. So we can see the lactate as the circle and the pyruvate. So we can see the two lines here easily. And the yellow part, as I said, was the part where there's the exercise, which that means, let me use blue here. That means this was before, the blue section here was before the exercise. And you use the purple here is after the exercise, what happens after. So we're kind of already playing and analyzing and looking at the graph. Now, after that, what can we see? We can see here, just by analyzing, let's look at lactate, which is a circle. So before exercise, it is low, right? So before exercise is low. During exercise, you can see that lactate starts to rise. And then after exercise, the lactate starts to drop, okay? And you can see that lactate rises a lot more than pyruvate. Now, what happens with pyruvate? Pyruvate is also low, and it continues to be low, but it's rising a bit, but very low compared to the other ones. And then, eventually, with time, it starts to rise more after the exercise. So you're kind of just looking and try to understand what's going on. So, from what I see here, you can easily see the lactate. During the exercise, lactate is in such a higher amount compared to pyruvate. That's what I got. And I can see that before the exercise, both of them are low. And after exercise, you can see that pyruvate is starting to rise while lactate is starting to drop. So I kind of did analyze. Before I even went to the question, I got more comfortable with the question, with the whole graph. Next is, what do these curves suggest? Now we have the options. Let's look at it. The first one, it says before exercise, so we're talking about the blue section, there was no pyruvate produced because there was no cell respiration. Now, pyruvate can be produced at any time, okay? And you're doing cell respiration at all times. So that would not make sense at all if we were thinking this way, right? It's impossible for us to not have cell respiration. Why am I saying this? I mean, we can have for a few seconds, and that's usually is when we're doing a lot of exercise that we don't have enough breath. When we don't have enough breath, then we can we can stop with aerobic respiration and go to anaerobic, where that's fermentation. But cell respiration is happening all the time because you need energy all the time. So all your cells are doing glycolysis at all times. So pyruvate is produced at all times. Remember that. So once again, with or without oxygen, pyruvate will always be produced. The question is, after pyruvate is produced, where are you going to go? As you can see here, in this process, up to this point, glycolysis is happening with or without oxygen. It is after that, once you produce pyruvate, that's when you know if after it you're going to go through aerobic or anaerobic. So A is already out of the way. B, it says here, during exercise, there was not enough oxygen available for the cell respiration. So that means during this yellow part, there was not enough oxygen respiration. Would that make sense? Well, for you to produce lactates, what does that mean? You need to not have oxygen. And you can see here on the graph that, yes, lactate is rising a lot, which that means there's not enough what? Oxygen. So far that makes sense. A lot of lactate is being produced because there's not enough oxygen. Makes sense. So the process was partially anaerobic. That makes sense because if you go towards producing lactate, that means there's no oxygen, and oxygen means aerobic. So B could be the answer, but we always have to look at C and D to see if there's more specific. C, during exercise, again, the yellow section, the level of lactate increased due to aerobic respiration. See, once again, lactate is only produced during anaerobic, so it's not produced through aerobic respiration. It's done when there is no oxygen, so it cannot be C. And then D, after exercise, so we're talking about the purple section here, the level of lactate decreases because there was not enough pyruvate to be used for anaerobic respiration. There is still pyruvate enough in there to go through the whole aerobic and anaerobic respiration. So it's not that the pyruvate is decreasing. This is because now what's happening is that you're starting to get oxygen back again. And once you get back oxygen, lactate go back to be pyruvate, and pyruvate continue towards the cryo-ATC. So remember, it will keep producing lactate as long as there's no oxygen. But once there is oxygen, lactate returns to pyruvate. So it has nothing to do with lactate going back to producing, not being able to be produced because there's not enough pyruvate. No, it's lactate returning to pyruvate, and lactate is becoming pyruvate because now there is oxygen. After the exercise, you sit down and you start breathing again. So again, the answer is B. So one of the things that comes from cell respiration is definitely they're going to put graphs or any comparison with photosynthesis. So it's very important that you know photosynthesis as well as cell respiration. So this question is combining 2.9 and 2.8, and I decided to put it here. So if you haven't done 2.9, it's okay, but I'm going to go through this assuming that you have learned photosynthesis, okay? So this experiment was set up to each test tube containing water of a pH of 6.3 and a pH indicator, okay? Now the test 1 and 2 also contain common pond autotroph. So when they say autotroph here, we're talking about there was a plant. So they put a plant on both. Now, carbon dioxide dissolves in water, right, and forms carbonic acid. We already know that CO2, if you place it inside the water, what does it do to the pH? The pH drops. So then it becomes more acidic. That's why it's called carbonic acid. Now, after 3 days, the 4 test tubes, so there's 4 test tubes, were found to have these results. So what do we learn from here? We have 4 test tubes. All right, let's put in another color. We have 4 test tubes, as you can see it. And if you look at them, all of them have a different pH. You have a pH of 7, of 5.5, 6.4, and 6.2. So there's 4 different pHs. And when it comes to, let's say, the night or dark, one is kept in the light, one is in the dark, one is a light, one in the dark. Okay, and now what do we have inside of them? We know that in 1 and 2, just the 1 and 2, there's a plant, right? These 2 here have plants. And this one, it just has CO2 with water only. But these ones have CO2 and water and the plants. So there's 3 things in these guys, and these ones only, these 2. So, again, analyzing the graph. Now, what conclusion can be drawn from test tube 1 and 2? Now, let's use the logic. How do we use the logic? We're going to use the logic thinking here about photosynthesis and cell respiration. Okay? So, if you have a bucket, and inside the bucket you got water with CO2 inside, right? And there is a plant here. Now, what will the plant do? Plant, they do photosynthesis. And when the plant does a photosynthesis, what is its reactant? It's CO2, right? CO2 plus light plus water, and then it converts into glucose. So basically it's using that CO2. Now, we know that the more you put CO2 in the water, the more the pH will drop. But if you get rid of the CO2, what happens to the pH? The pH rises. And you can see here that the pH has risen on this one, right? It's 7. Okay? Now let's continue with the rest of the topic here. What does the test tube and 1 concludes compared to these ones that don't have it? Let's check. First, the answer says photosynthesis has used CO2. It is true, right? It is true that both of them have used, they both have used CO2 because of photosynthesis. Second, test tube, respiration has produced CO2. Well, that's not true, because if you have plants here, what could possibly be have done? It would have mean that you actually have, again, for you to produce CO2, that means you have done cell respiration. Can the plant produce cell respiration? Yes, it could. But so we still gonna stay here. in like a question mark, right? Because yes, you can use for CO2 photosynthesis and you can do cell respiration to produce CO2. A plant can sometimes do this. And why is that? Because look, it says dark. So that means there's low light, which it's true. Look here, you see that it's light. For you to do photosynthesis, it has to be during the light in order to CO2. But be careful. We know also that photosynthesis has a section part, which is the second part, which is the light independent, which it uses CO2 to produce glucose without the need of light. So it could also be happening photosynthesis on it. So A is possible. Let's go for B. B here says photosynthesis has made the water more acidic. Why would the photosynthesis make the water more acidic if photosynthesis actually takes CO2 into the plant? If you take more CO2 into the plant, what happens? The pH will rise. So that means it will become more basic. And the photosynthesis does not make it more acidic. The only reason photosynthesis will make the water more acidic is if photosynthesis is releasing CO2. But photosynthesis is not releasing CO2. Photosynthesis is using it. So obviously B is wrong. C, I don't even have to look at this part. Photosynthesis occur but not in respiration. That's not true. Plants can do both. But at the same time, we don't know if cell respiration was happening, because if it was, the pH would have going to be dropping. So let's look at this one. Respiration occurred but not photosynthesis. No, photosynthesis happened here because you still can have the second part of the photosynthesis, which is light independent, in which the CO2 is used. So C is more likely to be a no. And then D, no conclusion can be drawn since pH in the controls has changed. No, it hasn't. So you can't figure out from this. So the right answer is A. A is more closely to anything. And again, just to reaffirm why, I'm going to show you why is A here. Because look, photosynthesis, there's plants in this too. So definitely they use CO2 because there's two sections, light dependent and light independent. These two are the two processes of photosynthesis. In the light dependent, obviously you're going to use the light and you're going to use the water and eventually you're going to produce ATP. And then the light dependent, you're going to use the ATP plus CO2, and you're going to make glucose. But all of this you need the plant. So you definitely could use in test tube 1 the light right here in order to make photosynthesis, and you have CO2, you can use it in order to make photosynthesis, so all good. Respiration has produced CO2. In this case, you could produce respiration because again, plants do make cell respiration. They're not just photosynthesis, but plants can also do cell respiration. So, and how can you prove that? You look at the pH. pH is very low, which that means the cell was releasing some CO2 as it's doing cell respiration during the darkness. So it's possible. So the answer is A. Now the cricket was placed in a respirometer. Now respirometer, as you can see, it's this whole thing here, right? All of this is the respirometer, which we're measuring the rate of cell respiration, okay? Now, they kept it at the temperature, so the temperature was constant, okay? So the temperature is not changing. And did this for 10 minutes, okay? And the soap bubble moved along the pipette. See, the bubble is right, oh, let me highlight it better. Like the soap bubble is right here, and it says that it moved. Now, which way would it move? If the pressure is coming, the cricket is right here. What is the cricket doing? The cricket is doing cell respiration, right? He's breathing oxygen. So he's probably breathing oxygen in, and then he's releasing CO2. And where CO2 is going? CO2 is going to the soda lime right here. It's being absorbed because what does the soda lime does? It absorbs CO2. So with that, what's happening as the oxygen is taken by the... So imagine there's a box, and imagine there is a lot of oxygen. These dots are all oxygen, okay? And you have the little cricket that I'm going to black here as a cricket. And then here you have under him something that absorbs the CO2. So every time the cricket is absorbing oxygen, there's less pressure in the atmosphere, right? And what happens to the CO2 that's releasing by the cricket is taken here, so it's absorbed in the bottom. So every time it keeps breathing more and more oxygen is going. So what's the pressure doing? The pressure is dropping inside. If the pressure is dropping inside, that means this is pushing the pressure that way, and that means the bubble is moving in this direction. So what does this show? This shows how fast the bubble is moving in this direction, and knowing the distance and the how fast it's going, that minutes, you can see how fast is respiration. So the question is saying, what was measured by the movement of the soap bubble? So the question, the answer that we have here is production of carbon dioxide, the volume of excretory products, which means such as P, the oxygen consumption, and the release of heat. Obviously, it cannot be carbon dioxide, because if it was the carbon dioxide, this would have, the production of CO2 will be coming out of it. But since we don't have that, we are actually looking at the amount of CO2, the amount of oxygen that is here, right? Is the oxygen that we were taking from here, from outside, because the oxygen, the CO2 was being absorbed, so it was not in play. So the right answer here is oxygen consumption. So like I said, it was all about the cricket respiring all the oxygen in the air, while the CO2 was being absorbed by the soda lime. So the pushing of the bubble in this direction here, it's because there is less pressure of oxygen in the air, and that causes the bubble here to move towards the cricket. Okay, because the cricket is still asking for more oxygen, so it can keep breathing more oxygen. So the right answer is C. Next, the image show a portion of a cell containing a mitochondrion. Okay, now where does glycolysis and electron transport occur? That's why one of the most important thing is that you have to know how to look at the electrographs or micrographs, in this case, of a mitochondria. Now you can see that there is this highlight in the corner here, and this is usually what we call the cell membrane, not the cell membrane, sorry, the mitochondria outer membrane. And then you can see the line is in the inside, and these usually are the cristae. And you can see the black dots here, which are basically the ribosomes. So they even have the cristae showing here. Now, they're asking, where does glycolysis happen? Now, glycolysis, let me give you this so you can see. Now, glycolysis usually happens in the cytoplasm. So the only one that you can see that is outside the mitochondria is this. So it can only be S. Now, is there any S here? Yep. And it's obviously already gave you the answer D right there, so it doesn't have to be work. Remember, because glycolysis, it happens in the cytoplasm. And the electron transport chain, it usually happens in the inner membrane, right? In this case, you can see it's not P because P is on the cell in the outer membrane. And then it cannot be R because R will be the matrix. So we only have Q here, and the Q is right on the dot of the cristae, so it's Q. All right, next question. What is the requirement? That means what do they need for accurate measurement? That means you want to make sure you measure it the right way, the rate of respiration using respirometer. So what are the things that you need in order to make this respirometer work? Now, first one says A, CO2 must be absorbed by water so that only oxygen production is measured. Now, this is just going back here to showing the picture right here. Let me just open, let me make it bigger here. They're talking about this, right? They're talking about this guy here. They're saying, what are the things needed, okay? You can see in this equipment that you have the mesh to separate the cricket from the soda lime. You have the soda lime to absorb the CO2. You have the stopper here to not allow oxygen or any other gas in or out. And you have the pipette to measure the soap bubble go in and out. And this is basically to see how oxygen is being consumption. So if an oxygen is being consumption, it's not its production, it's consumption. And CO2 is not absorbed by water. As you can see, it's not water, it's actually soda lime. Okay, that's one thing. Next, the temperature must be kept constant so the volume changes are only due to oxygen use. This is more likely to be it. See, because if you go back here, right? And it did say the temperature is constant temperature. So why? Because if temperature can manipulate the whole thing, so we don't want the molecules to move in any kind of faster pace. You want to keep it standing. So B can be one it, but let's look at the other ones. Here it says, oops, the anaerobic organisms are better used than aerobic ones as they will produce large volumes of changes. No, see, the whole point here is about oxygen. And anaerobic, is not using oxygen. And going back here in the picture, you can see that this is about getting the oxygen, right? So if you put a cricket that does not, is going through anaerobic, that means it's going through fermentation, which means it does not use oxygen. So what's the purpose of this whole molecule, this whole process here, is if it's to see the animal picking up the oxygen. So when you say here that the anaerobic organism is better, well, like, how is it better, man? You have to actually respire the oxygen to see how fast cell respiration is happening. So it's useless. And D, warm-blooded animals are better used than cold-blooded animals as they do not change the temperature. See, this is not about the blood and about the heat released by the animals. This is about respiring. That means it's about breathing oxygen. Well, that means warm-blooded or cold-blooded, they both going to go through cell respiration. They both need energy. So they both need oxygen production, oxygen consumptions, and CO2 productions. So of all of these, B is the closest one because we know that the temperature can manipulate the volume and the pressure of gases. So the temperature must always stay constant. Remember this, every time you're doing respirometer, it has to be constant. So it's B. All right, so now we're going for the next question. This next question is, again, there's a lot of times in IB exams, they will mix photosynthesis and cell respiration in one question. So here we have the final products of photosynthesis and aerobic respiration. Now, if you remember aerobic, that means it's oxygen, which that means we're producing ATP after the link, the Krebs cycle, the ETC. Photosynthesis, you're actually using CO2 to produce oxygen and glucose. So, photosynthesis, we are talking about product. Now, if you remember this, you should remember the equation. The equation is CO2 plus water with light will give you glucose, right, plus water and oxygen. Now, this is photosynthesis. Cell respiration will be this way. So it will have glucose with water and oxygen will turn into water and CO2. So this is back and forth. So photosynthesis, when we're doing photosynthesis, let's put it here, instead of the light, I'm going to just put P here, for P for photosynthesis and cell respiration here. So it needs products. The product should be glucose. Do we have glucose in this one? No, so it's canceled. This one? No, it's canceled. So it has to be B or C because it has glucose and oxygen. Do we have glucose and oxygen? We do have this. Do we have water? Yes. ADP? Nope. Because in the end, ADP will be in the glucose. So B would have been the right answer. But just to make clear, let's see on this side. And aerobic respiration, CO2 is the product, which that means we're going this direction. So yes, it is a product. Water is a product. And ATP, yes, it's forming ATP for energy because cell respiration is all about breaking down glucose here in order to give you ATP. So yes, B is the right answer. And it couldn't be C because it says carbon dioxide ADP. Not true. When you're doing cell respiration in this direction, you're not making ADP. You're making ATP because cell respiration is about breaking down glucose. And when you break down glucose, you're taking that energy. So the answer is B. And here's one more question related to cell respiration. And it's the respirometer, which is something very common is they put this equipment, again, same place. So diagram shows a respirometer used to measure respiration rate. Respiration rates of seeds. So right here we have plant. Plant is this case is not the cricket, is actually a seed. So plants can also respire. A lot of students think that photosynthesis is only in plants and cell respiration is in animals and other organisms. That's not true. Plants can also cell respire. That means they also can breathe oxygen in and release CO2, sometimes depending on what they're going through. So with that in mind, we have here Y. If you remember Y, it has to do with absorbing CO2, right? Because you only want to have oxygen in the area. And these will have to do with oxygen too. So in this right here is going to actually measure the oxygen movement. So let's look at what we have here. What is expected direction of the movement of the fluid at X? That means is the X going to move this way or is the X going to move that way? And the function of the fluid at Y. What exactly is the Y? If you remember, Y is about absorbing CO2. Let me show you back on the picture here so you can see what I'm talking about. You see that Y is the solder line, which is right here in the bottom, and this is what's absorbing what? CO2. Because you don't want CO2 to manipulate with the pressure, you just want oxygen to manipulate with the pressure in the air. So we already know that Y has to do with absorbing CO2. So if that's true, then this one is out and this one is out. So we only have, basically, let's put all of this out. So it's going to only be A or C. And it's funny because it's going to say, this is absorbing. Now, is this going up? Is this the right answer? Or is it C? Is it going down? Now, remember, if plant is here and is absorbing oxygen, oxygen is going inside of them and CO2 is going in here, what's happened to this area? This area is becoming empty. So if it's becoming empty, right? If there's nothing in there, what's going to happen? You have to bring new oxygen into it. So the pressure is coming out and pushing it. So that means it's going up. So the right answer is C. That means, remember, every time you're emptying this little guy here, right? If this area that I'm putting dots here, imagine these are the oxygens. If I start to taking away these oxygens, that means I need to fill new pressure into it. So that means it's coming in here to fill up the more oxygens in. So that's what it is. Every time you take more oxygen in, basically the manometer here is going to start moving towards the area that there was no oxygen, because the oxygen was taken by the plant or by the animal. So once again, the answer is C. And that's the kind of questions you can get for cell respiration for the SL section. Thank you.